Assalamu alaikum. Today we are going to study topic 1, that is cell structure and organization from the course Biology 5090. So let's start. I'm sharing my PPT. All right. So let's start. Bismillahirrahmanirrahim. Cells. What are cells? Cells are the basic unit of life, basic structural and functional unit of life. The body of living organism is consists of cell, and the cell consists of living matter which is known as protoplasm. The fluid present inside the cell is known as protoplasm. Basically, protoplasm it consists of nucleus and cytoplasm. So this jelly-like substance we say protoplasm. Here it consists of about 70 to 90% of water. The rest of the fluid consists of mineral salts, organic compounds like carbon-containing compounds that includes carbohydrates, fats, and proteins mainly. And its composition varies from animal to animal and from plant to plant. So see, this is a generalized structure of a typical animal cell. You can see here that this is the outline showing the cell surface membrane. The fluid present inside the cell is known as cytoplasm. Cytoplasm is a combination of basically two words. Cyto means cell and plasma means fluid. So whatever present in between the nucleus, this is nucleus, and this cell membrane is cytoplasm. And nucleus and cytoplasm together, they are known as protoplasm, as I mentioned in my previous slide. So they together known as protoplasm. So this is a generalized structure. And here the 3D structure showing the internal parts, the organelles, permanent structure present inside the cell in cytoplasm. So this is the 3D structure showing the nucleus, and these are the endoplasmic reticulum, Golgi apparatus, mitochondria. These are the mitochondria. Single is mitochondrion, plural is mitochondria. This is the cell membrane. Here you can see. So this the whole fluid present inside the cell membrane, inside the cell is cytoplasm. So obviously, under light microscopy, these structures they are visible. Okay, now the two main types of cells, prokaryotic and eukaryotic cells. Prokaryotic cells. Prokaryotic cells, it includes bacterial cells basically. So this is the diagram that is showing a bacterial cell. This one is the bacterial cell, prokaryotic cell is the same thing. So there are many structures which are absent in prokaryotic cells. As you can see here, no nucleus is present. And yes, this is the main feature, the striking feature of prokaryotes or bacterial cells, that they do not have nuclear envelope. That is, their DNA or genetic material is not enclosed inside the nuclear envelope, nuclear membrane, and it is scattered in the cytoplasm. Okay, you can see here that this is the flagellum, which is a locomotory organ of the bacteria. Similarly, here additional wall, cell wall is present, but it is not made up of cellulose as we will study in plants. Similarly, there are few ribosomes. You can see what are they. We will study later. Plasma membrane is present in all types of cells, and this is a generalized structure. Okay, now yes, this is a structure of a bacterial cell in a magnified view. So you can see here, yes, this is the loop of DNA, the main DNA, and there are small plasmids, structures called plasmids present here in the bacterial cells. So they are the structures which are present in prokaryotic or bacterial cells and are not present in all the other cells. So what makes the bacterial cell unique is that they do not have nuclear envelope, means they lack very basic structures. You can see only ribosome is present here and in the cytoplasm, right? And plasmids and this DNA loop is present, all right. And the other structures which are absent, you can see here that they are not present in the under the light microscopy. So this is about the bacterial cell, and now the parts, the structure of the bacteria. As I have shown you in the diagram, the cytoplasm is present, and it's again the jelly-like substance and contain very limited organelles. And yes, it is surrounded by a plasma membrane or cell membrane, and it contains very few organelles, permanent structures like ribosome is present, like DNA loop is present, plasmid is present. So these are some structures which are present in the bacterial cells. Similarly, plasma membrane is a partially permeable membrane. We will study about this partially permeable membrane in detail, and it's present around the cytoplasm. It encloses the cell and it prevents the cells from escaping. We will study about the function of cell membrane, that the main function of the cell membrane is to control what is entering and exiting in the cell. Similarly, circular DNA is present, as I have shown you in the previous slide, in the center without nuclear envelope. The genetic material is not enclosed in a nucleus, and there is a large loop of circular DNA. And what is the function of this circular DNA? It controls the cell division, it controls the cell development, and the cell activities going on inside the cell. The plasmids, small loops of circular pieces of DNA, they are also present, and I have shown you one in the previous slide. They contain genes. Genes are the hereditary units, and we will study about genes later in the upcoming topics. Then we have what are the function of genes, that they carry genetic information to help the process of the survival, that is reproduction in the bacteria. Bacteria, they are able to reproduce because of these genes, these hereditary units genes. Then we have ribosomes. I have shown you that they are scattered. They are the small round structures present in the cytoplasm. And the main function of the ribosome is protein synthesis. Cell wall is present, which is a tough and non-living layer, and it is not made up of cellulose as in plant cells, and it surrounds the cell membrane. Means this is an additional membrane other than the cell membrane present outside the cell membrane. Now, what is the function of the cell wall? It prevents the cell from bursting and allow the water salts to pass through, because it's a freely permeable. Its pore sizes are large, and it allows all the substances, mainly all the substances, to pass through it. And you can compare it, this cell wall, with the cell membrane, that it is a partially permeable membrane, right? But it's a fully permeable membrane. It's a fully permeable membrane. Okay, now, yes, next slide. Now, what are the characteristics of cells? So some characteristics I have already shown when we studied the prokaryotic cell, like plasma membrane. There are so many different names. Plasma membrane is also known as cell membrane. It's also known as partially permeable membrane. There are different names like selective permeable membrane. So these all names can be used by the examiner when the examiner is asking for plasma membrane. Protoplasm, I have shown you that cytoplasm plus nucleus, we call it protoplasm. Organelles, they are the permanent structures which are present in the cytoplasm. And control center is nucleus, which is the control center of the cell. All right, now protoplasm, as I told you, it consists of, it encloses each and everything, right? So cell surface membrane, cytoplasm is a fluid where most of the cell activities take place and many organelles are present inside it. Nucleus is present in all types of cell except for the prokaryotic cells and virus. And the fluid present inside the nucleus is nucleoplasm. Nucleus membrane is known as nuclear envelope or nuclear membrane, and there are one or more round structures present inside the nucleus which are called nucleoli. And chromosomes or chromatin network is also present inside the nucleus that contain genetic substance. Nucleus, what is the function of nucleus? It controls the cell activities, and yes, it's vital and essential for the cell division. All right. Okay. Now, nucleus, as I have told you about the nucleus, that let me show you the diagram. Yes, so this is nucleus. So this is the 3D structure showing the nuclear envelope. Here you can see the boundary of the nucleus that separates the nucleoplasm from the cytoplasm outside. And this is the structure nucleolus. Single is nucleolus, and when they are present more in number, so we use the plural term nucleoli. This is the chromatin network, the thread-like structure present in between the nuclear envelope and the nucleoli. And you can see that from outside, the nucleus is attached with the endoplasmic reticulum. These are the nuclear envelope is porous. You can see the pores present here in the nuclear envelope. So this information that whatever I have shown you is present here in this slides. As I have mentioned, nuclear envelope is a double membrane that surrounds the nucleus, and it separates the nuclear content from the surrounding cytoplasm. As I told you that it is perforated, that is, it has pores. And I have shown you some pores in the diagram as well. And thousands of nuclear pores are present, and they allow the materials to move into and out of the nucleus. Nucleoli, plural term is nucleoli, and I have shown you nucleolus in my diagram. These are the spherical structures, and the main function of these structures is the production of ribosomes. Ribosomes, if you remember, they are the structures present in the cytoplasm, and they are involved in protein synthesis, as I have shown you in the prokaryotic bacterial cells. Now, chromatin. Chromatin is a network of long thread-like structures and DNA bound to protein. Chromatin, it consists of the nutrient of the substance called DNA, and it contains the heredity information, heredity material. It controls the activities of the cells. We sometimes call the nucleus, as I told you, as a brain, as a functional unit of cell. So during cell division, these chromatin threads, they condense and become highly coiled structures called chromosomes. So in the topic genetics and inheritance, you will study about chromosomes in detail, about DNA and chromosomes, genes in detail. So this is a structure. And here you can see the nuclear envelope, nuclear membrane in a magnified view, showing the pores. And there are thousands of pores, as I've mentioned. It's a double layer, and you can see that the pores, they are very clear in this mount. Okay, now cytoplasm, what are the structures present inside the cytoplasm? So the first structure that we are going to study, which is present in plant cells, is chloroplast. Chloroplast. It's present only in plant cells and in few other different types of cells. Some bacteria, they have chlorophyll present in them. and some other type of groups of animals, they have this chloroplast present in them. Okay, so chloroplast, what is the function of chloroplast? It is a site where plant make its food using carbon dioxide from the air and water from the ground in the presence of sunlight. And you must be familiar with this process. This is photosynthesis. So chloroplast is basically the structures which conduct, which is a site of photosynthesis. It uses carbon dioxide and water from the environment of the plant, and you, in the presence of sunlight, this process it takes place, and glucose or sugar is formed. So this is the site where photosynthesis or this process takes place. All right, now ribosomes. So ribosomes, as I've mentioned earlier, that ribosomes, they are involved in protein synthesis. Proteins synthesis take place, and it's packed with proteins forms in the cells. They are packed with ribosomes. There are two different types of ribosomes: free floating, which are present freely in the cytoplasm, and the other type of ribosomes are attached with the endoplasmic reticulum. Here in the diagram, you can see this is the 3D structure of nucleus, and outside the nucleus, these are the endoplasmic reticulum. So these ribosomes are attached with the endoplasmic reticulum or found freely in the cytoplasm. Okay. Here you can see these are the ribosomes, magnified view, and this is the nucleus, and here is the endoplasmic reticulum showing the attached ribosomes at its surface. Okay, now vacuoles. Vacuoles are present in both plant and animal cells, and they are basically the fluid-filled space enclosed by a membrane. The animal cells, they may have small vacuoles, and they are not permanent. They are temporary structures. Means when the extra or excess food is available, so they are formed, and when they are used up, so they are disappeared. They are not the permanent structure in animal cells. And they may contain water and food substances, as I've told you. Okay, the plant cells, they have a large central vacuole, and it occupy a larger space of a plant cell. Inside the vacuole, the liquid is present, which is called cell sap. And cell sap contain the dissolved substances like sugar, amino acids, and mineral salts. The large vacuole is enclosed by a membrane called tonoplast. So this is about the vacuole. And yes, this is a diagram showing the central vacuole in plant cell. As I mentioned that it occupies the larger space. This is the plant cell, and the larger space is covered with this central vacuole. So as I've mentioned that it contains water, food, and also the waste. It in plant cells, this vacuole, they make the cell firm by swelling, and this helps the plant cell to maintain the support and rigidity. As you know that there is no skeleton or hard structure present in plants. So if you see, the support and the rigidity is provided by the central vacuoles in cells, in plant cells. Then we have mitochondria. Singular is mitochondrion and plural is mitochondria. Mitochondria is present in all types of cells, in plant cells, in animal cells, and they are the site of cellular respiration. Respiration is a process by which plant and animal cell, these different types of cells, they intake energy, they produce energy. And The matrix contain enzymes which break down the carbohydrates and other nutrients for energy. So this breakdown of the molecules in the presence of oxygen is known as aerobic cellular respiration. So mitochondria, the function is aerobic cellular respiration. Why we call aerobic cellular respiration? Aerobic means because it takes place in presence of air. Air contain oxygen. So because it takes place in the presence of oxygen, and here inside the cell, it is going on. So this is known as aerobic cellular respiration. And the energy releases maybe it's temporarily stored in small molecules called ATP, adenosine triphosphate, which are considered as the battery of a cell. So ATP can be transported from one part of the cell to another, and when needed, ATP breakdown to release energy. So this is about the mitochondria, but from the examination point of view, you have to focus that these are the structures. Mitochondria are the structures present in all types of cells, in plant cells and animal cells, and they are involved in cellular respiration or aerobic cellular respiration. Whatever the energy they form, it is stored in the cell in the form of ATP. So these are the points that you need to consider when the examiner is asked you about what is mitochondria, what is the importance of mitochondria. So this is that you have to write in the answer. All right, now the next slide showing the structure of the mitochondria. As I've mentioned, this is the outer membrane. Although it is not in detail, means uh, only the structures in the light microscopy. Here you can see mitochondria. The detail of the structure is not included in our syllabus. All right. So plasma membrane or cell surface membrane, as I've mentioned, that it separates the inside of the cell from the outside the environment. And what is the nature of cell surface membrane? It is partially permeable. Partial means some part, not full. So basically, it has pores in it, and these pores they allow the small substances only to pass through them. And this is why we call it as a selective permeable membrane also, because it allows the selective permeability. Yes, cell surface membrane control what is entering and what is leaving the cell. So all the cell activities going on inside the cell, it's controlled by the plasma membrane or cell surface membrane. Cell surface membrane, it is made up of lipids and proteins. So you can see. All right, now plant cell wall. Cell wall here in plant is made up of cellulose, a very tough material that consists of cellulose fibers. And cell wall, it protects the cell from mechanical damage and help the cell maintain its rigidity, its firmness under the Turgor pressure. So basically, this cell wall, it prevents overexpansion of the cell when water diffuses into the cell. And it is a barrier to many fungi, bacteria, and other organisms. And these organisms, they may cause diseases. So that's why this barrier is very, very important to inactive or to control these organisms. As I've mentioned that it's fully permeable, the pore size of this membrane is larger and it allows almost all type of substances to pass through them. Bacterial and fungi cells, they have cell wall, but it is not made up of cellulose. In the bacterial cells, we have seen the cell wall, and the other types of cells called fungus or fungi, they also consist of cell wall outside the cell membrane, but it is not made up of cellulose. This is the characteristics of only plant cells, that their wall is made up of a very tough polysaccharide, which is known as cellulose, a kind of carbohydrate. Now, the difference between cell membrane and cell wall, and it can be asked, the examiner can ask on 4 or 3 marks. So you must mention these points whenever you are comparing the cell membrane and the cell wall. Cell membrane present in all types of cell. As I've mentioned, this is the structure that is present in all different types of cells, in bacterial cells also, and in also cells of virus. Although in virus cells, we will study that there are many structures which are not present in virus. So cell wall present only in plant cells, in bacterial cells, in fungi, and in algae. By nature, cell membrane, as I've mentioned many times, it's semi-permeable. Semi means half or partially permeable. That is, it's allow only some substances, some small molecules only to pass through it. While cell wall, on the other hand, is fully permeable. The main function of the cell membrane is to protect the protoplasm, while the main function of the cell wall is to protect cell membrane also, and to also maintain the shape of the cell. Okay, so metabolically, cell membrane is alive, while cell wall is non-living and inactive. Cell membrane is made up of lipids, protein, and carbohydrates, while cell wall, in different types of organism, it is made up of, its composition may vary. As in bacteria, most of the bacteria, it is made up of a substance called peptidoglycan. In fungi, it's a different substance, and in plants, cellulose. So basically, this only cellulose cell wall is present in our sabers, that the name you need to mention. Otherwise, the composition of cell wall in fungus or in bacteria, it is not asked. So these are the comparison, the differences. Now, the difference between plant and animal cells. So plant cells and animal cells. Okay, so this question can be asked in terms of points, bullets, and in the form of diagram also. The examiner provides an outline of a cell, and then they ask you to draw some structures which are present in plant cells. So if you study the point, the comparison between the plant and animal cell structure, so you will be able to draw as well, and you will be able to write as well. So both can be asked. So let's study what are the differences between animals and plant cells. So plant cells, they have cellulose cell wall present, while here in animal cell, this cell wall is absent. Presence of chloroplast, this structure, the green color pigment present inside this structure, chloroplast, and this is present in plant cell only, while in animal cell it is absent. The large central vacuole is present in plant cell. There are numerous few vacuoles present in animal cells, and they are temporary as compared to the plant cell. It is a permanent vacuole and a large central vacuole. Cytoplasm reduced to a thin lining in plant cell, while cytoplasm fills the cell entire. So let's continue. Yes. The presence of starch grains in plant cells. The starch grains, they are usually formed from the glucose. Glucose is stored in the form of starch. All right, then here no starch grains are present in animal cells. Basically, these are the first 3 main points, are the main differences that the examiner asks. Now the, yes. Now the specialized cells, tissues, organs, and systems. All right. So these are the 3 specialized cells which are present in our syllabus, in the syllabus of O level. Root hair cells. These are the root hair cells. red blood cells. These are the RBCs or red blood cells, and the xylem vessels. Here you can see. So, what are the specialized cells? The first thing you need to understand that in the title here, specialized cells. What are the specialized cells? So, specialized cells are adapted, are the cells they develop changes in their structures to their functions. As you can see, this is a plant cell. This is a plant cell, right? But it is not like a typical plant cell. In a typical plant cell, as I have shown you in the previous slides, that they are consist of a, they have this large vacuole, they have this cell wall, the nucleus is present, right? But this elongation, this extra protrusion is not present. So here you can see that root hair cells, they develop this elongation of the plant cell because of the function, the task they are assigned. Similarly, this animal cell, here you can see RBC is an animal cell, but it lacks nucleus. You can see that there is no nucleus inside the RBCs. So obviously, it developed this change in its structure to its function. We are going to study obviously the other structure adaptation as well. Here, these are the xylem vessels. Xylem vessels, they are also, different. They do not have any protoplasm inside, and that's why they are known as dead vessels or dead cells, consist of dead cells. Why? Because there is no protoplasm. All right, no protoplasm. The living matter is absent inside it. So obviously, the function of this adaptation is according to the function that are assigned to these xylem vessels. So let's see what are the functions and the what adaptations. So first of all, the animal cell, only one animal cell is included in our syllabus, and RBCs are there. RBCs, the main function is to transport oxygen. As I've mentioned when we were studying the mitochondria, that mitochondria is involved in respiration process. So respiration, it requires oxygen. So the oxygen is transported cell to cell through RBCs. Inside the RBCs, oxygen is carried and it's transported to the oxygen poor cells or tissues. So what are the adaptation in the structure of RBCs? The RBCs, they are circular and biconcave shape. Circular, as I've shown you in the slide. And what is a biconcave shape? Here you can see this structure is the biconcave structure. So if you observe the RBCs from side, so they are appear like this. That is, they here you can see they are broader from the edges and they are somewhat thinner in the center. So what is the advantage of this adaptation, this changes in the structure? They are able to change their shape so that they can pass through the narrower blood capillary. This biconcave shape allow them to pass through the narrower blood capillary. Okay, then they contain hemoglobin. What is the function of hemoglobin? The hemoglobin is a red color pigment. As in plants, green color pigment is chlorophyll, which is present in chloroplast. So in RBCs, the blood appear red in color because of this hemoglobin, red pigment. So this hemoglobin basically it binds oxygen and it transports it to the oxygen poor cells. So absence of nucleus is the main structural feature of the RBCs, that they do not have nucleus. And what is the advantage of this feature? Why they do not have nucleus? Because they have to transport oxygen, and for that they need space. So RBCs, they have hemoglobin, and hemoglobin is bind with the oxygen and then it is transported to the oxygen poor tissues all around the body. Then we have xylem vessels. Xylem vessels and root hair cells, they both are plant cells. They present in plants. So the xylem vessels, the main function of the xylem vessel is to transport water along with the mineral salts from the ground through roots to stem and then to the leaves. Obviously, leaves are the structures, leaves are the organs that require water for photosynthesis. Photosynthesis takes place in leaves. So obviously, water from the ground is transported to these xylem vessels uh, to the leaves. Now, what are the structural adaptations? Changes in the structure. The absence of cross wall, there is no cross wall present in the xylem vessels, and protoplasm is also absent. As I've mentioned, that because of this absence of this feature, absence of protoplasm, they are considered as a dead cells. And if you look into the xylem vessels, they are like this. See, there are no cross walls. This is, if we consider it's a cell 1 and cell 2, so these are the partition walls that separates one cell from the other. They are absent in the xylem. And because they are absent, so they make a continuous channel to, the water and the mineral salt is passed. So basically, this is the advantage to transport the water fastly and rapidly. This continuous and long channel is required. Yes, another adaptation in the xylem vessel is the presence of lignin. The cell walls, they are strengthened by the lignin, a very tough material. Cellulose itself is a very tough material, and it also have lignin. So xylem, they are woody because of this lignified wall. The another type of cells are the root hair cells. The function of the root hair cell is to absorb water and mineral salts from the soil. So, what are the structural adaptation in them? They have long and narrow root hair to increase the surface area to volume ratio, to increase the size of them so that more water and mineral salts can be absorbed. As I have mentioned that in the previous slide that they have, yes, here, this is the long and narrow structure which is present in the root hair cells. So there are thousands of millions of cells present in root, and this helps them to absorb more water along with the mineral salts. Now moving toward tissues, organ, and system, the concept. So cells of the same type may be grouped together to carry out the special functions, and when cells are grouped, they form tissue. So tissue can be simple or complex. Simple tissues are made up of same type of cells, while the complex tissue, they are made up of a combination of different types of cells. As epidermal cells, they are consisted of similar type of cells. Epithelial tissues, they are also made up of the same type of cells. So they are the example of simple tissues in animals. And in complex tissues, the examples is nervous tissues and glandular tissues. We will study in upcoming topics of biology that what are the structural adaptations, and how these complex tissues are made. These are the four different types of tissues. Now we have organ. So tissues combine together structurally and functionally to form organs. As for example, stomach and liver is an example of organ in human. Leaf is an example of organ in plants. And you can see here in the diagram in torso that different organs are shown here. Then organ, it consists of organ system. Many organs, they are combined structurally and functionally to form an organ system. So the examples are, for example, digestive system, respiratory system, and organisms have few to many organs. In plants like stems and leaves, they make up a system. Human have about 40 organs that are grouped into about 10 named systems. The human nervous system, for example, includes brain, spinal cord, and nerves, network of nerves. So this is the digestive system. Digestive system consists of different organs starting from mouth and end at anus. And other than this, liver and stomach, liver and pancreas is not shown here. They are also present as an accessory organ. Okay, now level of organization. So in a body, as I've told you that body, the structure, the simplest unit of life is cell. So cell, they are combined to form, suppose that if these are the muscle cells, muscle fibers, so they are grouped together to form a tissue. Here you can see is a tissue, right? And different types of tissues are present. These are the circular muscles, these are the longitudinal muscles, these are the glands present in them. So different types of, it's a complex, basically it's a complex tissue, stomach. Then they all different types of tissues, they combine together structurally and functionally to form the stomach, which is an organ. And stomach along with the other organs like small intestine, like mouth, like rectum, like esophagus, right? So these all organs make up the digestive system. So this is the level of organization that cells combine together to form tissue, tissue combine together to form organ, and organ combine together to form an organ system, and organ systems, they make up an organism, a complex organism. So these are some examples of human body systems: respiratory system, digestive system, nervous system, blood circulatory system, excretory system, immune system, and so on. Skeletal system, system of bones, muscular system, system of muscles, reproductive system, different in females and male individuals, integumentary system, that is skin system, lymphatic system that filters the bacteria and destroy the bacteria, endocrine system. So these are different systems present in human body. In plants, the vascular tissue system consists of xylem and phloem, which are the transporting tubes. Now, the next part is the magnification topic and related to the microscope. So for now, I'm ending this class. We will study this topic, magnification and microscopy, different types of microscopy in the next class, inshallah. Take care. Allah Hafiz.